So you're all aware of the effects of brain stimulation on neurons. So I'll be talking about astrocytes, which, sorry, Carly, but these are the major glial cells in the brain. So astrocytes are dynamic modulators of neuronal function. So the bottom left figure here just shows the primary and the fine branches of astrocyte processes. So this allows a single human astrocyte to cover up to 2 million synapses. And this allows astrocytes to regulate synaptic transmission and support neuronal plasticity. So astrocytes are also integral in the response to central nervous system injury and have shown functional and dysfunctional roles in various neurological and psychiatric disorders. So given that electric fields don't discriminate between brain cells and the important role of astrocytes in plasticity, injury, and disorders, our group wanted to investigate the effect of rTMS on astrocytes. So there's been a steady progression of research illustrating the effect of brain stimulation on astrocytes in various experimental models. So most studies applied rTMS at threshold level, which makes it harder to distinguish direct effects versus indirect via synaptic plasticity or other downstream mechanisms. So I delivered low intensity subthreshold rTMS for my experiments, and I'll be presenting a subset of the data from my PhD for the remainder of the talk. So first, we wanted to examine the effect of rTMS on the astrocyte response to neurotrauma in a mouse model. So this was achieved by delivering a penetrating cortical stab injury to one hemisphere. So this injury is confined to the cortex. It doesn't elicit motor or sensory deficits and is practical for focally applying low intensity rTMS to the cortex. So the injury has a well-characterized astrocyte response where they undergo cellular and molecular changes, such as they proliferate, they swell, and they produce inflammatory molecules that can chronically persist at the site of the injury. So mice received 14 days of rTMS daily from 24 hours after the stab injury. And as the rTMS was low intensity, I was able to perform stimulation on awake, freely moving mice, as seen in the image here with the coil above the mouse head. So after the 2-week period, the brains were collected for immunohistochemical analysis. And regarding the stimulation protocols, mice received either sham above the injury site, 1 Hertz above the injury site, 1 Hertz contralesional, and the complex high frequency stimulation paradigm called BHFS, or biomimetic high frequency stimulation, which is used in our lab and is based on endogenous neuronal firing patterns. And it's previously shown success in correcting abnormal visual circuitry and upregulating BDNF in mice in our lab. So while 1 Hertz contralesional is common in clinical applications, we wanted to test the effect of high and low frequency stimulation directly to the astrocytes. And just to remind you that the low intensity rTMS that was applied is orders of magnitude below what evokes motor responses in clinical rTMS. So we also wanted to explore differences between male and female mice of different ages with respect to the rTMS, as hormones influence the astrocyte response to neurotrauma, and the progression and outcome is actually more severe with advanced age. So in this experiment, there were male and female mice of 3 months of age and 18 months for a total of 80 mice. So after I confirmed the consistent lesion size across the groups of mice, I examined the astrocyte response by counting the number of astrocytes around the injury and the volume actually occupied by astrocytes across the brain surrounding the injury. So on the left here, we actually have the injury site of the needle, and the astrocytes labeled in green with GFAP, which is the most commonly used marker of reactive astrocytes after injury. And then just here on the right is just a figure showing a higher magnification of astrocytes and more proximal to the injury site. So I also measured the expression levels of growth inhibitory molecules secreted by astrocytes after injury, shown here in red. Additionally, I measured the changes to macrophages and microglia, which are the brain immune cells, but I won't be covering these results in the interest of time. So I found no change in the volume of astrocytes surrounding the injury and no change to the inhibitory molecule expression between sham and rTMS groups, but there was a difference in the number of GFAP positive astrocytes surrounding the injury. So this graph shows the density of astrocytes for each group for each rTMS group in the adult and in the aged male and female mice. So on the left, we have the young mice, and there's the females and the males, and each rTMS group here is color-coded. So statistical analysis indicated there was a significant effect of stimulation and also the interaction between stimulation and the mouse age and the mouse sex. So if you look at the graph from left to right, the post-hoc analysis indicated that within the adult female mice, the astrocyte density was reduced following BHFS, the high frequency protocol, and in the adult male mice, the low frequency increased the density of astrocytes. And moving on to the aged female mice here in the 18 months, the 1 Hz stimulation reduced the astrocyte density compared to sham, and in the aged male mice, the BHFS increased the density. So we have this trend of direct stimulation affecting the astrocyte density at the injury, but in a sex-specific manner. So the density always went up in males, but down in females. But we weren't really sure whether the changes in the astrocytes were direct or indirect via neuronal modulation. So in an attempt to answer this, I moved to an in vitro model and removed the non-astrocytic cells from my cell cultures. So here are my astrocytes here on a cover slip here in the bottom left, again stained with GFAP in green, and you can see the nucleus of the cells in blue. So our lab has also designed custom low intensity coils for in vitro application where they can fit underneath a well in an incubator or below an imaging chamber. So these coils are miniature versions of commercially available human-specific coils, and they don't generate additional vibration or heat to the system. So these coils delivered the equivalent low-intensity subthreshold magnetic field strength as the in vivo coils from the previous experiment. And we also added this time CTBS and BHFS, CTBS and 10 Hz to the stimulation protocols. So sham 1 Hz, CTBS 10 Hz, and BHFS. So an in vitro injury model, the scratch injury, was performed on the cover slips to imitate a neurotrauma-like response from the astrocytes on these cover slips. So they do a similar thing where they swell, they proliferate, and they migrate over the scratch site. And we applied daily rTMS to the astrocytes after the injury, but we saw no changes in the proliferation, no change in the migration after stimulation, but we saw a reduced hypertrophy following 1 Hz stimulation at 1 day after the injury. So also our lab has previously shown that low intensity rTMS delivered at high frequency increases calcium levels in culture neurons. And we wanted to see whether rTMS altered astrocyte calcium. And this is a major determinant in astrocyte functions as it relates to intracellular and intercellular activities. Most notably is probably the modulation of neuronal activity. So I imaged the changes to astrocyte calcium using the ratiometric FURA2 calcium indicator. And I measured the calcium levels before, during, and after rTMS of various stimulation protocols. And each group consisted of 17 to 34 cells across multiple cover slips from 4 culture replicates. And these graphs just show the 1 Hz increase the intracellular calcium levels in the astrocytes during stimulation in the cytoplasm and nuclear compartments. And in order to try and isolate the source of calcium, the experiment was repeated in calcium-free media on the left column, where the calcium stores were depleted by thapsigargan on the right column. So the effect persisted of the 1 Hz increase in the calcium-free media, but was not present when the intracellular stores were depleted, indicating the source of 1 Hz increase was primarily from intracellular calcium stores. So we have a change in astrocyte calcium during rTMS, but we couldn't infer functional consequences from these changes due to the extensive range of functions that astrocyte calcium signaling modulates. So in order to get a better grasp of the downstream consequences, I examined gene expression changes 5 hours after stimulation in the same cultured astrocyte model. And I also tested protein level changes at 5 hours and 24 hours after stimulation. And these were collected from multiple biological replicates across multiple experiments. So this time around, we reduced the complexity of all the frequencies to assess only 1 Hz and 10 Hz against sham, but we included a second 10 Hz to allow for a pulse duration comparison in the analyses. So the extracted RNA was run on a qPCR array of 125 genes known to be influential in astrocyte biology. So this Venn diagram here shows the genes that were significantly downregulated by 2 to 4-fold times in comparison to sham for each stimulation group. So the genes affected by the 10 hertz are in the top 2 circles. So we have the 600 pulses, 10 hertz, 6,000 pulses, the 10 hertz. And the bottom circle here is the 1 hertz. And in total, there were 21 significant genes. So I don't expect anyone to know the function of these genes. I've color-coded their main function here in the astrocytes. And interestingly, I only saw significant gene downregulation following rTMS. And majority of these genes were related to inflammation and calcium signaling, which fit nicely with the previous experiments findings. And importantly, no upregulated genes. So I immunostain 4 of these genes for changes in protein expression, but in the interest of time, I'm just going to cover 2 of these proteins. So these are the representative images of 2 proteins in astrocytes at 5 hours and 24 hours after stimulation. So in green here is the STIM1, which is involved in the detection of depleted calcium stores from the endoplasmic reticulum. And this is shown here in the left graph here in green. And in red is ORI3, which is a calcium release activated calcium channel on the plasma membrane and is shown on the right graph. So I assess the intensity of immunolabeled protein, which is a semi-quantitative measure that I obtained from mean pixel intensity values from max projection Z stacks on a confocal microscope. So each bar on the violin plot here represents a different frequency group, and the split down the middle just represents the 5-hour and the 24-hour timing. So sham is on the left, and we have the 2 10 hertz followed by the 1 hertz groups. So at the 24 hours, I'd just bring your attention to those for the 2 things. We see a reduction in immunolabeled protein intensity for both of the 10 hertz protocols for both proteins, which complements what we found with the mRNA results, which is promising. And interestingly, the 1 hertz, which did not show a significant mRNA fold change, showed increases in the protein immunolabeled intensity. But 1 hertz was the same protocol where I observed increased intracellular calcium in my previous study during stimulation. So potential discrepancies between the mRNA and protein results could include the turnover rates of the proteins and the time points that I've assessed here for the gene analysis and protein analysis. So I suppose to conclude, my results highlight that rTMS can affect many aspects of astrocyte biology, such as calcium signaling, gene expression, protein levels, and a response to injury. In addition, these changes can occur during stimulation and in the hours to days and weeks following stimulation. So interestingly, we see aspects that are similar to neuronal effects, such as frequency selectivity for responsiveness and effects. But it is, I suppose, unwise to place a binary label such as excitation and inhibition on astrocyte activity because they don't behave in a binary fashion like neurons. So the next step is to determine whether rTMS changes in astrocytes affect neurons, and if they contribute to inducing network level plasticity within rTMS in order to better understand the effects of brain stimulation on the cells of the brain. So that's all from me. I just want to acknowledge my supervisors, especially Jenny, Roger, and Alex Tang, and the rest of the lab members and the funding bodies. And thanks to Jenny for chairing the session, and thanks especially to Alex for organizing this symposium session, highlighting the importance of the non-synaptic rTMS mechanisms. So thank you.
Thank you, Darren. That was a really great coverage of a lot of information, and astrocytes are just such interesting cells. So it's fascinating to hear about it. We have a question from the attendees. Hi, Darren. Thanks for the talk. This is from Athena Stein. I don't have any experience with animal models of TMS, so apologies if my question is a bit left field. there's an increased risk of seizure in human moderate severe traumatic brain injury compared to mild TBI, traumatic brain injury. I was wondering if your mice ever experience seizures as a result of TMS, given they have a rather severe brain injury.
Yeah, that's a really good question. No, our mice didn't. I had all them. I stimulated all the mice personally myself, and they never experienced. I never observed them experience any seizures at all. But you have to remember that we were stimulating at low intensity, so this was, you know, orders of magnitude below an intensity that could induce any, I suppose, any motor responses, any activity to induce seizure-like responses. So I think in other animal models that are done at higher levels, you can do super threshold to induce seizure activity.